purpose of our experiment was to use chromatography to investigate what types of dyes are present in non-toxic watercolor paint and red Kool-Aid. We will compare the two to see how similar the dyes used are and thus determine how safe the paint is. Our independent variable was the substance that we used for chromatography, either watercolor paint, one of the FDA-approved dyes, or red Kool-Aid. Our dependent variable was the RF values that we found for the different substances. In our experiment, we were specifically looking for red 40 dye in the watercolor paint and Kool-Aid. The molecular makeup of red 40 dye is C18, H14, N2, Na2, O8, S2. The sulfur trioxide groups, carbon monoxide group, and carbon projecting off of the main body of the dye give it its unique color in comparison to the other azo dyes.
Before we began our experiment, we laid out all of our lab materials, placed associated materials together so we would not lose anything, and ensured that everything would work. We closely followed our procedure and changed it when it did not work. We began by mixing our solutions of Kool-Aid and water paint, while the other one of us prepared 3 samples of chromatography paper. Then we used a micropipette to make 3 different sheets of chromatography paper. That way we'd have 3 trials to average. We put the Kool-Aid, the watercolor paint, and 7 FDA-approved dyes onto the chromatography paper, and then put it into jars with a small amount of solvent and covered it so it could develop. When it was done developing, we placed it on paper towels to dry, and then we measured things. Unfortunately, the watercolor paint did not show up, so we were unable to measure that. Later on, we decided to do another trial of watercolor paint. However, we were out of red paint, so we had to do it with orange paint instead.
After conducting our chromatography, we found that the red 40 dye traveled on average 2.9 centimeters. The blue 1 dye traveled 7.3 centimeters. Yellow 6 went 4.0 centimeters. Red 3 traveled 1.0 centimeters. Blue 2 went 2.9 centimeters. Yellow 5 traveled 5.1 centimeters. Green 3 traveled 6.0 centimeters. And the red Kool-Aid dye traveled on average 0.9 centimeters. We were not able to visibly observe how the watercolor paint dye traveled up the paper. Our solvent front traveled 9.0 centimeters.
After measuring the distances that the dyes traveled, we calculated the RF values of each. For red 40, the RF value was around 0.32. For blue 1, it was around 0.81. For yellow 6, it was around 0.45. For red 3, it was around 0.11. For blue 2, the RF value was around 0.32. For yellow 5, it was around 0.57. And for green 3, it was around 0.66. For the red Kool-Aid, we found our RF value of 0.1, and we did nothing for the red watercolor paint. We discovered that there is red 3 in the red Kool-Aid, and then when we did the second trial of watercolor paint, we found that it had yellow 6. Using the data we gathered from our experiment, we were able to determine several things. First of all, we found that the watercolor paint that we tested had FDA-approved dyes in it. We were able to determine this because the dye that we found in the paint had RF values that were extremely close to those of the FDA-approved dyes we tested. Also, we found that the concentration of dye in the paint was extremely low, as they didn't even show up until we concentrated it significantly. Our hypothesis was partially correct, as there were FDA-approved dyes in the watercolor paints. However, there was not nearly as much dye in the paint as we had expected to find.
While completing our lab, we discovered several random and systematic errors. First of all, the original concentration of the paint was not concentrated enough and did not register on the chromatography paper. We ended up fixing this error by redoing the chromatography of the paint with a significantly higher level of concentration. A random error was that we attempted to use a micropipette to put the dye on the chromatography paper, and it ended up leaving very large blotches of dye versus small ones. These large blotches of dye made our results less accurate. We could have fixed this error by using a toothpick or similar device to apply these dyes.
The results of this lab show several important things. First of all, it shows that the dyes used in non-toxic watercolor paints are FDA approved, and also that they are present in significantly lower amounts than they would be in stuff like drink mix. So at least in terms of dyes, watercolor paints are indeed non-toxic for children.